This method can help answer key questions in the antimicrobial peptide field about whether peptides localize to or translocate across the cell membrane. Using spheroplasts and protoplasts allows the visualization and analysis of peptide localization in a physiologically relevant model system without the resolution challenges encountered with bacterial cell imaging.
Though this method can provide insights into antimicrobial peptides, it can also be applied to other membrane-active agents, such as cell-penetrating peptides.
Gram-negative E. coli spheroplasts and gram-positive B. megaterium protoplasts should be prepared in a biological safety cabinet or laminar flow hood using appropriate sterile technique. If desired, bacterial strains can contain an antibiotic-resistant plasmid, and spheroplasts and protoplasts can be prepared at an open lab bench. In this video, spheroplasts and protoplasts are prepared at an open lab bench using antibiotic-resistant bacterial strains. To set up an overnight culture, use a sterile micropipette tip to transfer a single bacterial colony from a solid agar culture plate to a 14 milliliter culture tube containing 2 to 3 milliliters of 3% trypticase soy broth, TSB, for a 16 to 21 hour incubation at 37 degrees Celsius with shaking. To prepare gram-negative E. coli spheroplasts, dilute the overnight culture at a 1:100 ratio in 25 milliliters of TSB in a 250 milliliter flask, and return the bacteria to the shaking incubator for another 2.5 hours.